Okay, so welcome to all those who are coming in. My name is Destiny. I'm the NephCure social media and content coordinator. I handle all of our social media, but today I am joined by Dongwon Lee, and I will happily let him introduce himself. He is a part of Sampson Lab and also has his own lab. But Dongwon, could you start by telling us a little bit about yourself and your lab?
Sure. Hi, I'm Dongwon Lee. I'm an assistant professor at Boston Children's Hospital and Harvard Medical School. I'm a computational biologist and geneticist by training. My original training is more on the engineering side, as I received PhD in biomedical engineering from Johns Hopkins University. But then I became interested in human genetics during my graduate study and ultimately decided to pursue my postdoc training in human genetics. So after completing my training, I joined Children's Hospital in 2019 as an instructor in Dr. Lee and Sampson Lab. Now I run my own lab, as you said, and we have many research projects related to computational genomics and genetics in the context of human diseases. And of course, kidney disease is one of my main research areas.
Amazing. Thank you so much for sharing that very nice introduction. To kind of deep dive more into more of your research, what kind of inspired you to pursue research in genomics, genetics, and nephrology?
Yeah, so one of the most important research questions in genetics and genomics is to identify genetic cause of human diseases. And luckily, a computational method that I developed during my PhD work had a great potential in tackling this problem. I mean, I didn't really know the implication of my work at the time, but as I began to understand that implication, and then eventually drew my attention to the genetics and genomics, and I ultimately decided to pursue my research in that field. So regarding the nephrology, to be honest, I had not been exposed to it until the later stage of my postdoctoral training. I was involved in one research project where we studied rare kidney diseases in the context of the blood pressure regulation, like Gittelman and Bartter syndrome. And during that study, I learned how complex the kidney tissue is, and also learned that the genomics in nephrology is relatively less explored to other research area. But that was pretty much about it, my exposure to nephrology at that time. And Matt Sampson is the person who really got me involved in this area. So in 2019, I was looking for faculty position, and one of my friends introduced me to Matt and said that he was looking for a computational genomic person as a junior faculty in his group. So essentially that was me. So I talked to him several times and realized that there are so many things that I can contribute to nephrology and kidney diseases with my expertise. So once I realized the potential, it was actually quite easy decision for me, and here I am. Yeah.
Amazing. Thank you so much for sharing. I really do appreciate you taking the time to answer that in depth. What are some of the key research questions that you're currently investigating, and are there any projects that are related to nephrotic syndrome that you're working on currently?
Sure. So one of the main research questions that we have in my lab is to understand the genetic variation in regulatory elements in the human genome, and how these elements, variation in elements, contribute to the human diseases. So what are the regulatory elements? To talk about the regulatory elements, we first need to know what the gene is. The human genome contains roughly 20,000 genes, which are essentially blueprint for proteins. And proteins are actual functional components that do all the work in cells. So that's the gene. But less than 2% of the human genome are used for encoding these protein sequences, and the remainders have other function. For example, human genome also encodes the information about where these proteins should be made and how many of them should be made. the regions that determine these are called the regulatory elements, okay? And as you can imagine, mutations and genetic variants in these regulatory elements can increase or decrease the risk of the disease and may even cause diseases. The problem is it is still challenging to figure out where these regulatory elements are in the genome. And more challenging problem is to figure out which mutation or variants can impact the function of these regulatory elements. To tackle this problem, we use a machine learning and statistical approach, a lot of computational approaches per se. For example, in a recent study in collaboration with Matt, published in Nature Communication a few months ago, we have identified thousands of genetic variants associated with changes in gene expression in kidney tissues. And importantly, we also found that these variants are more likely to be associated with kidney function, such as estimated glomerular filtration rate, EGFR, or proteinuria. So what that means is that changes in gene expression caused by this genetic variation in regulatory elements can lead to the variation in functions in kidney, which can ultimately affect the disease development and progression. So that's one of the major findings. And so about the nephrotic syndrome, yes, the same logic can be applied to the rare disease such as nephrotic syndrome. In fact, there are many ongoing research projects related to the nephrotic syndrome in our group. So for example, another recent study led by Matt Sampson and his collaborators has identified many genetic variants associated with steroid-sensitive nephrotic syndrome. I was also co-author of that study. But we still don't know which genes in which tissue or cell types are these associated variants are actually exert their function. So we are now trying to figure out which of these genetic variants are impact the regulatory elements. So we have a specific hypothesis, especially in the kidney tissue and also some of the immune cell types. So and then there are others, but that's pretty much it.
Amazing. Thank you so much. And what are some of the major challenges faced in kidney genomic research today? And how are you working to address them through the research that you're doing?
Yeah, so from my perspective, I think the kidney, as you may know, kidney consists of many different cell types, right? And the proportions are very different. For example, if you take a piece of sample from kidney cortex, vast majority of them are probably proximal tubules. On the other hand, only 1% of them are probably podocyte, which is an essential cell type for kidney function, right? And these rare cell types are difficult to study simply because you don't have enough materials for any experiments. But even if you don't have enough data point to begin with, there are still things you can try as a computational biologist. So we recently published a new computational methods to maximize the discovery of the regulatory elements from these rare cell types using machine learning technology. So that's one example that we try to overcome this challenge. There's another ongoing project where we are trying to develop and optimize computational framework that allow us to effectively combine multiple data sets from different individuals. So by doing so, you can also increase the sensitivity of the regulatory element detection, especially for these rare cell types. Essentially, you're just pulling up and aggregate more data. But there are challenges in doing so because of the heterogeneity between the samples and individuals, which that you need to really overcome. So those are the type of work that we are working on right now.
Absolutely. Thank you. And I know you discussed some of the collaborations and co-authoring with some of the research that you've done, but what collaborations have you established with other researchers in the fields of genomics and nephrology? And I guess, explain how have these collaborations helped advance your research?
Yes. So the most of the research projects that we are working on cannot be done without collaboration, right? So genomics and genetics studies are becoming more and more complex, and I think requires expertise from many different fields. So although I have my own research group now, we are still closely collaborating with Matt Sampson's group on many different projects. I'm a computational biologist with a strong computational and engineering background. And I also have learned a lot about the nephrology over the last few years after joining this wonderful division. But there's still a lot more to learn. On the other hand, Matt is a pediatric nephrologist and also have a lot of experience in computational genomics and genetic studies, but that's not his major training, right? So our expertise and experience are very well complementary to each other, and thus it has created a lot of synergy in our collaborative work. So it's been working wonderfully in all research projects that I've involved so far. So, I mean, as you briefly mentioned, the project that I introduced in the previous questions are also, all of them are collaborative work with the Matt Sampson Lab. So I think really the expertise, I mean, these days, most of the research project requires expertise from different fields, and the expertise that I don't have are usually filled by collaborators such as Matt and others.
Thank you so much for sharing. And I guess the last question to kind of kick this off before we open up the floor to any other questions. What are some of the principles that you follow when conducting research, or any mantra that you sort of live by?
Okay, so I think I emphasize the data quality a lot in all of my research in general. So in computer science, there's a phrase, garbage in, garbage out. So it means that bad input will always give you bad output, okay? So in other words, no matter how great a method you use, it doesn't really matter if your data is of poor quality. So I always say that you should first check the data quality carefully, okay? There's another one. I also jokingly say this a lot. I don't trust anyone. Of course, there's a context to say this, right? So in science and research, we analyze the data received from other people, obtained from other projects, or use methods developed by other people. That happens all the time. In many situations, you will find that the description of the data or the methods is different from the actual contents that you analyze. So if you just blindly trust what is described on the surface of the data or method that you're using, you will often end up making mistakes and find different results. So I also emphasize this a lot, and I always put a lot of energy into learning the details directly from the contents. If it's method and then software, I tend to go directly to the source code to figure out what they implemented. And if I receive the raw data, if I have access to the raw data, for example, I always try to analyze from the raw data to make sure that, I analyze the data that I think.
Thank you so much. And thank you again for joining us for this kind of Q&A style interview. I really do appreciate your time and your knowledge and your research, because your research is definitely incredibly valued, especially in the nephrology space. As you said, I'm sure you need collaboration in this kind of space, as with any
Yes.
research. But again, thank you. I am going to open up the floor for a few minutes for any questions potentially, but if there are no questions, then we will share some links below in the chat for your research lab, the Sampson Lab, and other necessary resources that are going to be added below. But thank you. We'll take a few minutes to take any questions.
Thank you.